Assalamu alaikum students. Before going to our educational topic, please subscribe my channel Muntzir Mehdi Science TV on YouTube. Press the like button and share this lecture to your friends and classmates who have no access to it to add in their science education. And those students that are preparing privately can benefit from our channel very effectively. Let's come back to our topic, that is the second part of that video that I have made during the last days, namely the factors affecting the rate of reaction. So in this tutorial video we will conversation about the remaining three factors, that are pH, enzyme concentration and substrate concentration. And it is the second part of that video during which I have clearly described about temperature with the help of theory diagrams and a temperature graph. So students, now we study the pH effect at the enzyme's activity. That is the second point of factors affecting the rate of reaction. So secondly, we study about what is the pH effect on the enzyme's activity. Dear students, we start with pH is a scale upon which the numbers are written from 0 to 14. That represents the pH scale, which tells us that a specific thing is acidic or basic or neutral. So it is used to know about the acidity, basicity or neutral nature of a substance. Students, from here, 7 number to the left from 0 to 6 are representing the acidic pH. For example, if a substance is representing 2 pH, then it will acidic. But if it is representing 4 pH, then it will also acidic, but it will less acidic, because moving towards left, the acidity is increased. So acidity is gradually increased towards 0, and one number is most strong acidic, and this acidity is gradually decreased towards 6, and 6 number is most weak acidic. Similarly, the numbers from 7 to right side are representing the basicity of a substance. As we move towards right side, the basicity is increased. So 8 number is most weak alkaline and 14 number is most strong alkaline. So students, we can say that when the pH is lowering, then it is moving to most acidic nature. But when the pH is increasing, then the substance is moving towards most alkaline nature. So this has made our pH scale. Before the 7 there is acidic representing numbers, but after the 7 there are alkaline nature representing numbers. So we can say that in between acidic and alkaline numbers, the 7 number is left, that is representing the neutral medium. Dear students, often substances have their specific pH value. For example, human blood have a specific pH value. Pepsin enzyme also has its specific pH around 2 pH. Not only pepsin, but every enzyme has its specific pH value. Like trypsin is a alkaline medium enzyme. Saliva also has its specific pH. So there is a very deep relation between the enzyme and its pH. Therefore, the enzyme is totally dependent on the pH for its proper functioning, its proper activity. As every enzyme has its specific pH value more or less, upon which it shows its function excellently. If we change that pH more or less from that pH, then enzyme can't perform its function and its activity will be disturbed, because the pH affects the amino acid. Students, how? Let's see. Beforehand, if an enzyme's amino acid have positive charge, then by changing the pH, this positive charge can be negative or neutral. So it will cause a major problem, controversy with amino acid. As we have conversation in our previous video lecture that an enzyme consists of a large number of amino acids that are attached to each other with the help of bonds. Due to the presence of these bonds, Students, the shape of the enzyme is organized, but these bonds may break due to the change of pH, due to the difference in pH. Students, let's suppose this amino acid has positive charge, but by changing the pH, this charge will be disturbed and will change to negative or neutral. So a bond between the amino acid may break, that will lead to a process known as denaturation that I have described during the first point in my last video. So after denaturation, whole of the enzyme will be destroyed, change its shape, and will not be able to perform any catalytic activity. So the main effect of pH upon the enzyme is it will change the ionic property of the enzyme. That is a horrible issue. Students, so for the proper functioning of the enzyme, the pH must fix and stable so that the enzyme will work best. For example, pepsin enzyme can work best at about 2 pH. This 2 pH is acidic number. So pepsin can perform best catalytic activity in the acidic medium, and its 2 pH is known as optimum pH. Like optimum temperature, optimum pH is that pH at which an enzyme shows its maximum activity. Next, we talk about trypsin enzyme that is present in small intestine of the mammals. That is an alkaline medium enzyme and shows best activity in an alkaline medium substrate at a pH of about 9. It is also known by the name of chymotrypsin, that is a form of trypsin enzyme. Its pH is 7 to 8 number. So students, it acts at a neutral pH or slightly basic pH. Likewise, pancreatic enzyme lipase is alkaline medium enzyme whose pH is about 8 to 9 number. So it is a slightly weak basic enzyme. In this way, there are some other enzymes and their pH in the form of a table in the biology and zoology books, from which you can estimate the acidity and basicity of an enzyme if you want. Those pHs are the optimum pH for their corresponding enzymes. Students, but when the pH is too low, then hydrogen ions can combine with R group of the enzyme's amino acids. So this combination reduces the enzyme's activity, its ability to attach with the substrate molecule. So consequently, we can say that there are a large number of enzymes that are working at different pH values. Some of them are present in human body also, which are present in different parts of our bodies. Some enzymes can work in acidic pH, some can work in basic medium, and some show neutral pH. Students, if that specific pH changes, then the enzyme will affect and can't perform its catalytic activity ably. So similarly, by the change of very high temperature, an enzyme can denature. Likewise, by the slight change in pH, an enzyme can also denature, and the rate of reaction will be slowed down. Next, we talk about the enzymes concentration. That is a third point. So, in this point, we will see that how can the enzymes concentration affect the rate of reaction as well as the enzymes activity. To understand this point ably, consider these are our five substrates. To catalyze these substrates, we have only two enzymes, E1 and E2 only. Let's pause in just 1 second. One substrate will bind to this E1 enzyme and the second substrate will bind to this E2 enzyme. And in first second, only two substrate molecules will be catalyzed. Students, why? Because only these two substrates found the chance to be attached to these two available enzymes. So the rate is 2 molecules per second, and these two molecules in just 1 second is recent rate. Furthermore, if we increase the concentration of enzymes, because here we talk about the enzymes concentration. So if we increase the enzyme's concentration, now instead of 2, there are 3 enzymes, then 3 substrate molecules will attach to these 3 available enzymes and the rate of reaction will be 3 molecules per second. So now the rate of reaction is more as compared to the previous condition. So we can conclude that by increasing the concentration of the enzymes, the rate is also increased. Now if we provide some more enzymes like 2 enzymes, then all the substrates will be attached to all these enzymes active sites, and in just 1 second whole of the substrate molecules will be catalyzed and the rate of reaction will be 5 molecules per second. Students, at that time, if you think that by increasing the concentration of the enzymes always increases the rate of reaction, then you are wrong. Because not, it is not so. It has a limit. Therefore, a stage comes when whole of the enzymes are occupied by all the substrates. At that time, here will not have even a single substrate left. This stage is known by the name of saturation. So the definition of the saturation is as when all the available enzymes active sites will be occupied by all the available substrates, then this stage is known as saturation. So now all the enzymes have got saturated. Now the substrate is constant. So if we add some more enzymes, then the rate of reaction will not increase because the substrate molecules are the identical, semi. So the increase of the number of enzymes will not increase the rate of reaction. Students, as we have only 5 substrates, so if we add hundreds of enzymes, then the rate of reaction will not affect, not increase, and we will have only the 5 molecules because the number of substrates did not rise. So to a limit, the rate of reaction increases by the increase of the number of enzymes. But after that limit, the reaction can't increase. Which limit and when? The answer is that when the number of enzymes will become equal to the number of substrates, because there is no advantage by adding more enzymes. Next, we see the fourth point, that is substrate concentration. Substrate means and concentration means quantity, amount. Let's again consider this diagram. During this point, if we increase the concentration of substrate, because this point is of substrate concentration. So when we increase the substrate concentration, then what does happen? Let's see. Now we have 5 substrate molecules and 10 enzymes. So in first second, all the 5 substrates will be attached to 5 enzymes active sites. The remaining enzymes will left and the rate will 5 catalyzed molecules. After that, we have to increase the number of substrates. So if we increase 3 more substrates, then these 3 substrates will occupy these 3 enzymes and the rate will be 5 plus 3, 8 catalyzed molecules. Students, believe also, still we also have 2 extra enzymes that have empty active sites. For their empty active sites, we will again add 2 more substrates. Now these 2 added substrates will occupy the 2 living enzymes active sites, and after catalytic activity the rate will be 10 molecules in just 1 second. So by increasing the substrate number, now we have concluded that by increasing the substrate numbers, the rate of reaction increases. So now we are relaxed by all this activity that by increasing the number of substrates, the rate of reaction also increases. But next, see, our quietism will be finished, wound up, when by increasing the more number of substrates. So when we increase 2 or 3 more substrates in the system, then there will not happen any change because there is not present any empty enzyme to be attached. So the rate of reaction will not increase. Students, why? The reason is that the more number of substrates as compared to the number of enzymes does not cause any increase, any change in the rate of reaction, because that will not cause any advantage. The increased number of substrates leads to saturation, during which all the active sites of the enzymes occupied by the substrates, and there will not any empty enzyme. So when there will not left any empty enzyme, then the increasing number of substrates will not increase the rate of reaction, and the rate will remain the same. Students, here you can also watch yourself that by increasing the number of substrates, the rate of reaction is not increased, not affect, unless the number of enzymes are not abundant. So students, in first second the rate will remain the 10 molecules, and those that are the extra substrates, they will wait for the freeing of the enzymes. So we can conclude that by increasing the number of substrates, the rate of the reaction will increase, but only to a limit. At the limit time saturation occurs. During saturation the rate will remain constant. Constant means the rate of reaction will not increase. So students, in this tutorial video, we will conclude that the activity of the enzyme is affected by the four things. First was temperature. By increasing the temperature, the rate of reaction increases. But a time comes when by increasing the temperature to a very high quantity, the rate begins to decrease. And a very high temperature leads to the denaturation of the enzyme. Second point was pH, which affect the ionic property of the amino acids of the enzymes. So the bonds between the amino acids can break up and also leads to the denaturation of the enzymes. During enzymes concentration, by increase the concentration of the enzymes, the rate will increase, but after limit the rate will not increase but will remain the constant. Similar is with the substrate concentration. By increasing the concentration of the substrate, the rate of reaction will increase. But when all the enzymes will occupy, then the rate will not increase and also will remain the constant again. Dear students, I hope now you are truly and tolerably understood both of the parts of this video lecture. That is very important question for the annual examination and often comes during exams. So preparing it ably, because now you have a complete concept to write this topic in your own words. Although at the end, I want to encourage you that please take care of your parents, your elders, and yourself. Also, take care of all those persons that are associated to you in some way. Students, also spend some time with your elders, because they also have some right to you, and sometimes they feel a strong need of your time, of your association. So make your strong relation with them. Finally, many thanks for watching this your video.